In order to understand the entire reproduction process within the flower, it is necessary to understand the three events within which this reproduction process in flower is divided. The main events or the main three events that takes place for the reproduction are pre-fertilization events, fertilization events, and post-fertilization event. Various processes undergo each of these three events after which the entire reproduction process occurs. So let us first of all begin with the pre-fertilization event. In this event, the processes with the help of which differentiation and development of both the reproductive parts, that is androecium and gynoecium, occurs. So let us understand the processes with the help of which differentiation and development occurs. First of all, let us begin with the male reproductive part, that is androecium. Definitely, in order to understand the androecium part, it is necessary to understand about the structure of stamen. As said earlier, a stamen is considered as the male reproductive part of the flower. It basically consists of three regions, that is anther, the top portion, followed by connective, the middle, and joined to filament. Anther is a structure which contains pollen sacs that liberates pollen grain. With the help of which the fertilization process occurs. Now the number of stamens in each flower can vary, and so it is necessary to understand the internal structure of anther. As it can be seen from the figure, this figure depicts the internal structure of anther. Now, anther, as it can be seen from the figure, is originally a bilobed structure, but due to the longitudinal layers present within, it forms a four-lobed structure, or it appears as a four-lobed structure within. Therefore, the structure of anther is bilobed with two theca present in between them in each. Definitely in each lobe, which is formed by the longitudinal division or longitudinal layer formed between them. As a result, the structure of the anther from within appears to be tetrathecous that contains four pollen sacs. And each sac contains various pollen grains. Thus, as it can be seen from the figure, the pollen sacs that contains pollen grains dehisces in various ways through pollination and thereby the reproduction process starts. Thus, the anther appears to be as a tetragonal structure, that is, it possesses 4 microsporangia, which bears the pollen sacs. These are the microsporangia which bears the pollen sac which further contains pollen grains. And therefore stamens are called as microsporophyll due to the presence of microsporangia within these anther. As it can be seen from the figure which describes the transverse section of an anther and the microsporangia. As it can be seen from the figure, the microsporangia within the anther appears to be circular. And this microsporangia consists of mainly 4 layers, that is epidermis, the outer portion epidermis, the next endothecium, following middle layers, and the innermost, that is tapetum. The outermost layer epidermis, the second layer endothecium, following it the third layer, that is middle layers, and the fourth, that is the innermost layer known as tapetum. Moreover, along with this, as it can be seen from the figure, the center of each of the microsporangious tissue consists of homologous or homogeneous cells in each of the microsporangia. Such compactly arranged homogeneous cells are known as sporogenous tissue. Thus, sporogenous tissue occurs in the form of a group of cells or group of homogeneous cells which is compactly arranged within each microsporangia. Now, as it can be seen from the figure, the outermost layer, that is epidermis, is made up of about 3 to 5 layers of cell. Consists of 3 to 5 layer of cells and occurs in a stretched or flattened manner. The following layer, that is endothecium, is appears to be a fibrous layer. That is a layer which is made up of fibrous tissue. The main function of such a layer is to provide protection and help in releasing the pollen grains from the matured anther. Thus, along with providing protection to the inner layer, it also helps in the dehiscence of the pollen grains from the anther. Following comes the middle layer, which acts as the layers of jointing between tapetum and endothecium. Thus, these layers act as a jointing medium of endothecium and inner layer, that is tapetum. Which is followed by the fourth and the innermost layer known as tapetum. Tapetum forms the innermost layer, which occurs in a single-celled form. That contains dense cytoplasm and a consistent nuclei. The main function of this tapetum is to provide nourishment to the pollen grain. This figure describes the mature anther that contains anther sacs which liberates pollen in form of grains from the pollen sacs.